Okay, next speaker is Nicolas Deguines from the University of Poitiers.
Good afternoon. Thank you, Gabriel and Emmanuel, for inviting me to discuss about the Spipoll project. So I'm going to present you the project and its main results. Before to start, I'd like to acknowledge that it's not just me talking, it's all the collaborators on the project. So first, the participant of the Spipoll, the OP, which is an entomological NGO, so in particular Mathieu de Flores and Pascal Dupont, and Romain Julliard, Colin Fontaine, Grégoire Loïs, and many others. So I'm going to start with the objectives of the Spipoll and then present you how it works exactly, and a little bit of what changed since the beginning of the program. I will show you the main results and some scientific perspectives. When we talk about pollinators, often the first thing that comes to mind for the public, but as Danny showed, also for scientists or in the literature, we are thinking about honeybees, bumblebees, and maybe sometimes wild bees. There are reasons for that. There are many species of bees worldwide, and both adults and larval stages feed on nectar and pollen. So that makes them very good pollinators. And so most pollination ecologists would agree that bees are the predominant pollinator for plants and ecosystems. But bees are not alone out there. And this study, for example, was looking at pollinator frequency by groups and for different plant species. So in this pie chart, we have the visitation frequency for different groups. And bees here, it's only about a third of the visits to this plant. And most of the visits comes from non-syrphid flies, and then comes other groups as well. If we look at other plant species, that it will be a different set of visitors in different proportions. And the main message is that each plant is visited by a high diversity of flower visitors. And it's actually very difficult to know precisely for sure which is pollinating and which is just visiting without pollinating. And some works have shown or conclude that the frequency of visits on flower can be used as a surrogate for efficiency. And even if there may be some, maybe there is sometimes it's not working, but at least it seems important to consider all flower visitors. So it's not just bees, it's also other hymenopterans such as ants, wasps, and so flies. It's a lot of flies, butterflies and moths, beetles, and even spiders. There are a lot of crab spiders, for example, on the flowers. And with the Spipoll project, we include all of these groups. All these groups have very different life traits in terms of food needs, in terms of nesting habits, physiological response, volatilism, etc. And this can lead to differences in responses to environmental gradients, for example, and potentially various population trends. With the Spipoll, we aimed at having a national scale coverage because France is very diverse in terms of biogeographical regions and also in terms of habitats. Here I only show 3 broad categories that I will reuse again after: urban, agricultural, and natural. But anyway, it's very diverse in terms of habitats, so it's important to have data from everywhere, if possible. So to sum up, with this project, we aim at obtaining data on a broad taxonomic scope, keeping the interaction with the plant, not just the insect, but also the plant that the insects visit, a national scale, if possible, long-term survey in order to, in the end, get population trends, and having a non-lethal protocol as well for the observations. And this is done since 2010 by a citizen scientist. So how does it work exactly? In other words, what are they doing here? So it's based on citizen science. So very briefly, it's the involvement of volunteers in scientific research. So it's a collaboration between a scientific team and a community of volunteers working around on the same questions. So it's quite simple, but it's yet standardized. Anywhere in France, participant will pick a flowering plant species of interest, the one he wants or she wants. And then the aim is to photograph all insects that visit the flowers of that species. So in the end, you obtain a picture of the plants and the set of visitors that visited the plants during the session. At first, in 2010, we had two sampling effort options. We had a flash option, which is 20 minutes exactly of a photographic session. But we also allowed for a longer session, so we called it the long protocol. And so it allowed the participant to make observation during more than 20 minutes, and also to come back to the species, multiple times during 3 days. But since 2018, we stopped the long protocol just to increase the number of exactly standardized protocol and making sure we have more and more 20-minute exactly sampling efforts. After this session of photographing, the participant ends up with a lot of pictures of insects, and the aim is to sort them into morphospecies. There is a huge diversity of flower visitors, and identifying them is very difficult, as Denis just said. For example, for this one, you have to capture it, kill it, pin it, view it under a microscope, and look at a very specific detail. And we chose a different approach, so using morphospecies, defined as a group of species that differs from the other morphospecies in any external features that can be seen on pictures of live insects. To have this approach, we developed an interactive online tool, so it's just like a key, but it's not dichotomic. Here on the left, you have the different descriptors of the morphospecies, so different criteria, and people can answer them depending on what they can see on their pictures, and they don't have to answer all of them. But if you answer them all, you are more and more, you get more chance to end up with a particular morphospecies and to finalize your identification. This is based on the Expert 3 tool that makes a different kind of interactive tools. So about the morphospecies, just some information on their taxonomic resolution, because as you can imagine, it's not all species because it's based on pictures. So here you have the number of defined morphospecies in the online tool, the identification key. And here you have the number of defined morphospecies by taxonomic resolution. So it goes from 1 for a group of species from different families within word order, and until 7 for a single species, so a species strictly speaking. So for Hymenoptera, we have still a fair amount of species, but many morphospecies, are not defined at the species level. The same is true for the other main orders of flower visitors, and the order that is a little bit different is for butterflies, because many species can be potentially identified on pictures if you have enough pictures and potentially from both sides of the wings. In total, we have 630 morphospecies defined, so that's grouping all flower visitors in France. So they are all put into the 630 different morphospecies. And almost half of them are species stricto sensu, and for 70% of them, the genus is none. For plants, we are using now PlantNet, so I'm not going to say more about it because it's the next talk. And how it works after that, so people put online their pictures, and there is a need to validate the identification. For example, for this picture, the morphospecies is the black and yellow sawflies, and that was proposed by the author of the picture, so the participant. And in 2010, participants could comment on that picture and even emit doubt. So they would flag that picture like, maybe this is not sure. And entomologists from the OP would validate the identification, validate or change the identification. So that was a lot of work for these people. And that was changed for different reasons. And since 2019, there are what we call peer validation possibilities. It took a lot of time for experts to validate all the data. Also, the participants gained a lot of entomological skills with participation. And there was also a will to allow for new way of participating to the project. So not just collecting data, but also validating the data for those who are capable of it. And so now people or participants, they can validate the identification. And if there are more than three validations, then we consider that identification to be validated. And approximately, we have more than 95% of pictures that have received at least 3 validations by the other participants. So it works pretty well, and they are really happy to be able to contribute that way. So to sum up, we have data on interactions between plants and visitors. We have a location, we have a sampling effort, date and time, obviously, weather condition, and all of that is what we call the collection in those people. And this is how where they are distributed in France. So we have more than 70,000 collections in France since 2010. So some main results. So we have two kinds of results. We started by analyzing the responses of flower visitors to land uses, and especially urbanization. And more recently, we had some work on the consequences of participating for volunteers. So more social, let's say, research. But I'm not going to talk about this research today. Very basically, what we did was to combine data on pollinator data with land use data. And then we estimated the response or the changes in occurrences for different morphospecies according to the different land use types. We computed land use affinity indexes, so indicators of affinity for the urban, agricultural, or natural land use types for our flower visitors. And the idea was to answer that question: in which land use are found wildflower visitors at the scale of France, and are cities refugees, for example. So here on the y-axis, you have the urban land use affinity, and each dot here is a morphospecies of the order Hymenoptera. For those that are below 0, it's an urban avoider morphospecies, so it's less seen in urban areas than expected by chance. If a morphospecies is above the line, it's what we could call urban exploiters, and if it's overlapping with 0, either they are tolerant or we don't have enough data to know for sure. So if you look at Hymenoptera, in average, it looks like this order is rather tolerant to urbanization. But if we consider now the other orders, we have much more urban avoider morphospecies in these orders. In 2010, when I did the analysis, we had about 42% of the morphospecies analyzed that were urban avoiders. And recently, I've redone this analysis with the data from 2010 to 2022. So I could analyze 235 morphospecies. And out of these, we had 72% of morphospecies that were significantly urban avoiders. So if you sum up this graph, it's here again. So it's the affinity here for urban land use for the 4 orders: beetles, flies, Hymenoptera, and butterflies. And now I'm going to show you the affinity for the agricultural land use and urban land use. So of course it's a little bit different. The mean average affinity for agricultural land use is rather positive. So to me it's not surprising. I mean, it's more interesting for flower visitors because there are more resources than in urban areas. Also, again, for Hymenoptera in average, we don't have a significant signal. And for natural land use, it's particularly positive for beetles and for butterflies. Now on these graphs, the black dots are actually only the what we could call the common morphospecies. So common in the sense that we recorded them often in our observations of the Spipoll. So common means that they were observed in more than 2% of the collections made in at least one of the 3 land use types. And otherwise, they are infrequent morphospecies. That means that they were observed in less than 2% of the collections made in each of the 3 land use types. So this was made to correct for the bias we have in the sense that we have more collections made in urban areas, for example. So now we can see how they differ in their affinity for the different land use types. And for the affinity with urban land use, the infrequent morphospecies are more urban avoiders. They avoid even more urban habitats than the common morphospecies. There is no difference regarding the affinity for agricultural land use, and there is a difference for natural land use, so the infrequent morphospecies tend to have a more positive affinity for natural land use. And given the definition of the morphospecies, the infrequent morphospecies include more species in the taxonomic sense than the common morphospecies. One last result, very briefly. Here you have an analysis again on the 3 land use types, but the 3 land use types taken together and the interactions, the triple interactions between them. So the result will be shown on this ternary plot, and you have here urban proportion in the landscape, agricultural proportion in the landscape, and semi-natural proportion in the landscape. So landscape here means that we are in a 20% urban landscape, 10% agricultural area, and 70% semi-natural areas. And the color here in the ternary plot is the predicted richness, number of morphospecies within 20 minutes based on the collections made by the participants. So what we can see is that the maximum predicted richness is in landscapes with about 70% agricultural areas and 30-40% natural areas. And on the contrary, urban areas rather poor compared to the other landscapes. And actually, wherever you are on this ternary plot, so in whichever landscape you are, if you increase the urbanization, it decreases the predicted richness of flower visitors that you will get in your collection. So this paper goes into more details of the same plots, but for different groups of morphospecies, but I'm not saying more right now. So one might wonder, so why do they vary in their response to urbanization? So there can be many different reasons for the variable response to urbanization. And something we looked at is specialization toward floral resources. So on this graph here, you have the mean community specialization, so the mean specialization in the collection made by the participants. If you are at the top, it's a collection made of rather specialized species, and at the bottom, rather general species. And we are going to see what happens with increasing proportion of urban land use. And there is a decreasing trend, so that means that there is more and more generalist species in frequency in the collections made by participants in urban areas along the urban gradient. And this corresponds to a functional biotic homogenization with urbanization at the scale of France. So some scientific perspectives that we have. There is currently an ongoing study on the effect of pesticides on as much as possible flower visitors that we can study with those people, so we will see. So this is based on a new database of annual pesticide sales in France at a quite fine scale, geographical scale, because it's a city or a few cities. And it's annual, and it's the sales for each substance, pesticide substance, so it's quite precise. And if we link this with the toxicity of the substances, half-life, the metabolites, etc. Then we came up with an index, which we called, for now, a total hazard ratio. But we can also have information on a sort of a cocktail index because we have all these hundreds and hundreds of substances. I don't have any results yet, unfortunately, but what I can say is that some models are running for about 20, 40 species that we are able to identify on pictures. Some interesting perspectives will be, as I said at the beginning, to try to get some population trends. So we could try to begin to do that because we have 14 years of data, over 600,000 plant visitor interaction records, more than 4,000 participants, so that looks pretty great. But there is still some challenges. Actually, only a small core of participants are long-term participants, and that's an issue with most citizen science projects, unfortunately. But for example, we have about 4% of our participants that stay for more than 5 years. And there is thus a high turnover of participants, so 85% of them stay for only 1 year. So that's a huge challenge, and that's the case even though we have a newsletter, annual meetings, webinars, and a lot of things going on. So we still have to work on that. So about population trends, I have nothing more to say except that it's a perspective. It needs to be assessed whether or not we can do it with some certainties. Something that would be interesting is to actually get to the interaction networks level. We potentially can do it because we have the insects interacting with the plants. And in this paper by Estelle Renaud from the University of Paris-Saclay, she investigated plant pollinator networks available in the literature and defined at the species level. And she tried to see what was the influence of downgrading them. That means lowering the taxonomic resolution of the insects and also of the plants and also together. How that would impact the metrics of the interaction networks. So in terms of metrics, she looked at nestedness, connectance, modularity, etc. So different classic metrics looked at in interaction networks. And to sum up her findings, the relative values of the different indicators remain the same, regardless of the taxonomic resolution. So that means that if we stick to one taxonomic resolution, we can rank the different networks. And this is interesting because applied to the Spipoll, we could build interaction networks, plant visitor networks at the Spipoll taxonomic resolution, and assess how the metrics that we compute maybe change or not according to different environmental gradients. Such as urbanization, pesticides, different agricultural practices, also with time, perhaps. So, again, some perspectives. And finally, that's perhaps a question or a dream, I don't know, for me. But we could wonder if it can be expanded to other European countries. In France, we have 4 biogeographical regions, and these are present in most of Europe as well. But I suppose that's not enough because we should still make probably a lot of updates in the interactive online tools to include for new species added as there will be new species in Spain or in other countries. So there will be a lot of work to before expanding, I suppose. And the same for plants. Although we have PlantNet, so see next talk. I would like to thank you for your attention, again the participants and everyone who contributed to those people.